Thank you very much, Nancy. So one of the big difficulties we had, like Janet introduced this morning, is biochar is highly variable. One of the things I've been trying to do is find out what we've looked at in the past to try to guide our future. One of the areas I've been turning to is coal. And a lot of people first say, well, biochar and coal, what are you doing? These are the exact same materials. They're created under different processes, but the materials are the same. The thing I find interesting about all the coal research is this is one material that we have researched for over 700 years. We have a very good understanding of how to burn it to create electricity. Yet, we can't come up with a structure for coal. This is very fascinating, and it should be one of the things like a little caution flag of saying, wait a second now, if we've looked at all coal and we can't even come up with a structure, what is our chances with biochar? The overall conclusion from all of the coal research has been it's a function of the original feedstock, conditions of the diagenesis, burial conditions, and the post-creation exposure events. These are the exact same conclusions we've been seeing with biochar. So if we look to coals, they rank them by what they refer to as ranks of coal. This is solely based on the volatile matter content, which was also alluded to this morning. They found that this volatile matter is directly correlated with its BTU content. So then we have coals that range everything from the peats all the way up to graphite. The one thing I like pointing out to people is the different physical appearance of these materials. You wouldn't guess that they're related as closely as they are, but it's just a very small difference in volatile matter will change the physical appearance of the materials. So like I was saying, we have a very long history. It was over 100 years ago that Potter first discovered that fungal colonies will degrade coal. Galli in 1910 said, that's great, we have fungal degraders, but then we also have bacterial degraders. This then led to a whole idea in the early 80s, right after our last big oil crisis, that we might be able to liquefy coal. Instead of having a bunch of train cars carrying coal, we could liquefy it into a liquid fuel and pump it through a pipeline. So there was a bunch of research started to look at various fungal degradation pathways, if we could get actually a liquefied black energy product. Fukasa in 81 said, yes, we can achieve this just with the ordinary soil microbes. And then Cohen and Gabriel even found that white rot and brown rot fungi will accomplish this very well. And they were actually able to see droplets of this black liquid form on coal. So this then led to about the 1920s. We were getting very excited about the potential. There were soil scientists starting to convene and talk about let's use coal in agriculture because they started seeing some yield improvements when they applied crushed coal to fields. Lasicki in 1929 said, yes, we can apply it and it'll even stimulate microbial activity. Thompson in 33 said, yep, we should start using this coal and charcoal mixes in animal feeding. Very similar to conclusions that you find today. What happened? We had the Great Depression. During the Great Depression, why would anyone spread a valuable energy material to a cornfield? This then became practice. People forgot about it after the Depression, and now we are to the point we are today. One of the points I like making is they have really no fundamental idea of how coal degrades. Our best mechanism right now by Fukasa is called the ABCDE mechanism. And all it is identifying coal that has particular groups or particular components that they know is susceptible to attack. Yet they don't know how it goes about attacking, which again is kind of amazing considering all the research that has gone into it. So we have from this area a real long history. One of the most fascinating parts that I found was we started seeing that coal was disappearing. In the 1830s, there's a great report that I read that thought that they had robbers coming in at night stealing their coal piles. And what they actually found out is that they can lose 10 to 25% of their coal if it gets wet. It'll just disappear. Groves and Thorpe said, oh, yep, that's great, but we only see about 2% in moist air. And then in the Engineer, which was one of the very first publications of scientific methodology in London, they said, oh, but if we put charcoal, coal in salt water, it won't dissolve or disappear. They said that they stored some in 10 years, and we only lost 0.1%. Then Fitts and others in 1907 said, oh, this is all great, these different storage mechanisms, but it all boils down to what the rank and the particle size is of the coal. Bruce in 1940 was trying to come up with what guidance to have for the storage of coal in the UK. The only correlation that he found over all the studies over the last 100 years that he looked through was that the quicker the charcoal pile is made, after it's formed, it'll disappear quicker. So the rate of the coal and charcoal piling was an important part of how quickly that pile disappeared. So what does this all have to do with biochar? And why am I even talking about it? There are three simultaneous processes that we worry about with biochar weathering. The first is chemical. Biochar is reactive. We can chemisorb oxygen. It has self-combustion properties. There was just recently a lab out in Tennessee that was entirely burned to the ground after they made a 1-pound batch of biochar in their oven. They forgot to close it properly, and it self-ignited and burnt the lab down. It probably would happen in my lab, too, if we made biochar, so I stay out of that. It also is a very interesting compound that is exothermic when it absorbs oxygen and water. Why is this important? What is the first thing you grab if you see a fire? A bucket of water. If you have a charcoal fire and you throw a bucket of water on it, you will actually increase the fire. This has been historically why there's been so many charcoal fires and why they are so dangerous. Once it gets going, we can't stop it with water. So we have all those chemical ways. Now we also have microbes. And everyone's been looking at these two pathways that, well, biochar has to disappear by microbes or chemical, because that's how we think it is. I should note over on the bottom left, that's a little piece of corn stover with microbes attached to it and eating it. I have yet to see that in all the biochars that I've looked at in my lab. I've been trying to find in the literature a picture where there is a microbe physically eating the biochar. I see a lot of fungal, I see a lot of bacteria on it. I don't see them actually eating and leaving the marks of their eating spots. But I digress. So getting back to this, there's a third process. It's physical weathering. We have microbial, we have chemical, and then we also have this physical weathering process. This should have been the first thing soil scientists thought of, because this is how we get soil. Rocks break down into soil. This is the process that's occurring to biochar once it's present in the environment. So all of these things have been alluded to, but no one has really put all these pieces together yet. Hockaday in 2007, yep, we can have microbial-aided disintegration. This is what happens with concrete. It's why concrete starts breaking apart, why your concrete blocks don't last forever. All of these end up forming condensed aromatic compounds. Hockaday observed them in the waters from leaching from these various black carbon deposits. Jaffe in 2003 in his paper in Science said, yep, this dissolved black carbon gets into our surface waters, and it's a possible explanation for the overall black carbon paradox, or the disappearance. So like I said before, the compounds are reactive with water, and we have to remember that. So what did I end up doing? I ended up going into the man cave with some of my students that actually got trained to run this instrument. I'm just there for kind of comic relief. We looked at these both with an SEM and as well as elemental dispersion systems. The fancy things are we get to see pictures down at very small levels. So we started looking at, well, let's just take char and rinse it. What happens? What is actually going on? So all these are just kind of shown. We put it in, we mixed it, we filtered it out to get the char after rinsing. Particularly, we looked at that filtrate. People typically just put it into another instrument, push a button, get a number. What we decided to look at was what's in there. Let's evaporate that on an SEM mount. So what we find, this is a picture of a fresh biochar. And like's been mentioned, they have stuff on them. It's covering material. You see the oils, everything is kind of covered. After we rinse, we start seeing an entirely different spectrum. But then I started seeing other things. Do you see all the little fragments that are present on that rinsed biochar? All of a sudden, why are they there? What's happening? Are they under the oil? Is it being caused? Where are these originating from? So to give you a little better picture of this, it's a very chaotic picture. The actual structure of the biochar is just breaking apart into small fragments. That's not the only thing that happens. We have a lot of other structures end up forming on biochar. This is what I call my biochar sinkhole. What's happening here is there's layers of the biochar that are being eroded below it. And the top layer of the graphite sheet just falls in and collapses, like all the sinkholes you've been seeing on TV recently. We also have, due to that water expansion of the graphite sheets, they start expanding to the point where all of a sudden pieces start falling off. That's what we see here with the big chunk falling off of that sheet. So it has really been an interesting process thinking about all this. So as I was sitting there being the comic relief for my students on the SEM, I started thinking, where have I seen these before? I know I've seen some of these types of structures. Luckily, my geology hat came on. These are the same erosional features in the Goblin Valley. We have things that actually defy gravity. How did this structure actually get formed? Yet, I can find these on the biochar. It is fascinating to see these type of analogies. So in addition, we have a lot of areas where rocks mysteriously start folding. Now, whether these are from the pyrolysis, whether it's from weathering, I still haven't completely figured all that out. But we do see exact same erosional relic structures on the biochar. So what? Who cares? So it breaks down. We know it does. Every time you touch it, you end up with black fingers. This hasn't been quantified. All of a sudden, we started looking at, well, how much could be lost to this rinsing? We start seeing 1, 2, 5, 15% of the biochar disappearing into the water. People are, oh, well, how does that relate to what we've seen microbially and all of the oxidation? Those are less than typically 0.05, 0.01%. This particular study here was on those exact same chars. So the more fascinating story to this is what's happening in that filtrate, or the evaporated rinse solutions. We started seeing that they're colored. What's given them the color? We don't have very high DOC measurements from our persulfate oxidation. So we looked at those evaporites. They're evaporated onto the SEM mount. We start seeing micron and nanoscale sized particles of this biochar being present in that water that was evaporated onto the SEM. Why do we care about that? There's been a lot of work looking at the toxicity and what's happening with carbon nanotubes. Everyone in the biochar area should be aware of some of this literature. We look at this, and these are the mechanisms for getting compounds past cell membranes. We have seen that the uptake of plant roots and animal cells depend on the chemistry of these very, very small fragments. What have they done with all these carbon nanotubes? They have seen that it can increase plant growth. They've put in nano-sized particles of charcoal with tobacco, and they found tremendous yield improvements. The question comes down to is why? One of the things they found was there's an actual change in the genetic expression in these plants. To put it simply, it means that we're comparing apples and oranges when we look at plant growth. We have different expressions of genes. They are no longer the exact same species of plants, really, because these genes are their fingerprint. All of a sudden, we see that the aquaporins are increased when we have these very small carbon particles. People are like, oh, that we know is the water channeling. That's what creates larger stomata. All of a sudden, we start seeing a mechanism for why we see increased yields. This all can help in regarding to plant expressions to environmental stresses, if we know how to control and understand this mechanism. So overall, this biochar discovery is that it's prone to physical degradation. It depends on how much it's mixed. The carbon loss is from the biochar directly getting dissolved into water. This also helps this whole black carbon paradox problem that we've been having. Look at all the back of the envelope calculations of how much char material should still be out in our soils. This has one mechanism now for its disappearance. The one thing that I do want to bring up is this doesn't mean that, we don't want small particles. We might actually want these things to break down to smaller particles because it is through that stabilization and association with clays that these gain a helmet, that they're entirely protected, that char doesn't even see the environment anymore. This also improves their mechanical strengths. The amorphous materials we use in all of our airplanes today, those are all our carbon composites. We've known now we want an intermixing matrix. We don't want things ordered in sheets. These are some hints and guides that we should be utilizing. How do we produce a biochar with higher mechanical strengths? Because now that is the key to making a biochar to withstand these. So with that, these are the agencies that provided funding, and also like to thank the students that I have that actually allow me to get all this data. So thank you very much.